Okay, now we will start with the new chapter, that is apicomplexa. So we have already discussed what is apicomplexa. Apicomplexa is a phylum of parasites. They are put under the phylum apicomplexa because they all have a structure which is common, is called as an apical complex, right? So what are the parasites which comes under apicomplexa as Plasmodium, Babesia, right? Toxoplasma and Sarcocystis, Isospora, Cyclospora and Cryptosporidium. So these are the parasites which are under the phylum apicomplexa. Now we will start with Plasmodium. Okay, so Plasmodium, if we talk about, we have the definitive host for Plasmodium. Plasmodium causes what? It causes malaria, right? So Plasmodium causes malaria. The definitive host for malaria is all the sexual cycle will take place inside the mosquito. So for malaria, the definitive host is the female Anopheles mosquito. Why is the female? Because in the mosquitoes, the male will only feed on plant juices for their livelihood, whereas the female mosquito need blood for the sexual cycle to complete. So that is why only the female bites the humans and animals, whereas the males are not involved in the transmission. So the vector for Plasmodium is female Anopheles mosquito, and the intermediate host is of course human. Right? So infective form for humans in malaria are the sporozoites. Okay, the sporozoites which are transmitted from the animal to human. From the mosquito to human through the bite. And from the human, if the mosquito bites, what is the infective form for the mosquito? That will be the gametocytes. Okay, so it will be the gametocytes for the mosquitoes. So all these are very important MCQs. It has been asked many times in many exams. So it is MCQ. So they can twist and turn anything of everything. These 4 points they can ask you anyways. Okay. So you should be very thorough about the infective form, which is the diagnostic form, which will be the infective form for the vector. All these you should be aware of. So now we will move on to the life cycle. So if you're talking about the life cycle of the Plasmodium, what happens here? The mosquito, the mosquito bites a man, and then from here what happens? So the mosquito, when it bites man, it will inject the sporozoites inside the humans, right? So the sporozoites are injected. Now the sporozoites will directly go to the liver for the further development. So in the liver, that is also called as, this liver cycle is also called as the exoerythrocytic cycle. Okay, so this exoerythrocytic cycle, what happens in the liver cells? The sporozoites will further develops into, they will undergo the cycle again, like trophozoites, they will convert into cytosol. And this cytosol will rupture and it will release something called as the merozoites. Inside the cytosol, we have the cells that is called as merozoites. Okay, so these merozoites will be now into the circulation, and then it will go and affect the circulating RBCs. So in the RBCs, and this cycle is called as the erythrocytic stage. Okay, so this is the erythrocytic cycle. So what happens inside the RBCs? It will start multiplying, and in the RBCs, it will undergo, the first stage will be trophozoites. There will be early trophozoite, there will be a late trophozoite, and then they will form the cytosol. Again, it will burst and it will release the merozoites and the gametocytes. Okay, and then again it will start infecting the new RBCs, and the cycle will start, will keep on going. So here when a mosquito bites a man, this gametocyte form will be taken up by the mosquito, and inside the mosquito all the gametogenesis takes place. So the gametes will fuse, right, and then they will form the gametes will become, the zygote will become a ookinete, and then they further develop to form these mature sporozoites, and these sporozoites are infective to human when the mosquito bites. Okay, and now, so in case of a transfusion, suppose patient gets the transfusion directly from an infected person, so what will be the infective form? So the trophozoites are the circulating form, so the infective form in that case would be trophozoites. Okay, so there will be no exoerythrocytic stage. That means the exoerythrocytic stage are the hypnozoites. So in that case, there will be no relapse. Okay, there will be no relapse in such case. Fine, fine. So now, what will happen? So this you should know about the vertical transmission and the blood transfusion. Now, what are the different species of Plasmodium causing malaria? There are 5 species which is causing malaria. One is Plasmodium vivax, Plasmodium falciparum, Plasmodium ovale, Plasmodium malariae, Plasmodium knowlesi. Okay, so these are the 5 species of Plasmodium that causes the disease. And also you have to remember few terms for this. Okay, so the every cycle inside the RBC, depending on that, they are given few names. Okay, so this will be benign tertian. Okay, this will be malignant tertian. Ovale will be ovale tertian. This will be quartan, and knowlesi is quotidian. That means this RBC cycle will take place inside Plasmodium vivax for 48 hours, for falciparum also 48 hours, for ovale also it's almost 48 hours, for malaria it is 72 hours, for knowlesi it is only 24 hours. That means what happens is, so once one RBC cycle is over, that means when the full complete development inside the RBC is over, it's going to burst, right, and release the merozoites into the circulation. And that is where, when you get the clinical symptoms, when the merozoites are released into the blood. So that is why the names are given. So every 48 hours, the cycle of RBC is completed and one set of parasites are released into the blood on the 3rd day after 48 hours. Right, so that is why it's called tertian. It is benign is given name because vivax is milder form compared to the falciparum. So this falciparum is given as malignant tertian, and ovale is ovale tertian. And for malaria, the RBC cycle takes 72 hours, 3 days to complete. So every 4th day, they will have this burst of RBCs and the merozoites are released into the circulation. On every 4th day, they will have the symptoms. So that is why it is called as a quartan. And knowlesi is just 24 hours. So every 2nd day, they will have the symptom. So it is called as quotidian. Right? Fine. So this is about the different species of Plasmodium that is causing the disease. So now there are 2 terms which you should know. One is called as relapse. So relapse is basically because of the dormant forms. So that is the hypnozoites. So the species which are causing relapse are Plasmodium vivax and Plasmodium ovale. So how to remember this? I used to remember it this way. Relapse is comparative to recrudescence is smaller alphabetical letter, right? It's a smaller letter. Similarly, among the Plasmodium species, vivax and ovale are the smaller ones. So remember for relapse, it is vivax and ovale. For recrudescence, the bigger names. So now, this recrudescence is caused by the presence of the parasite in the peripheral circulation because of inadequately treated. Okay, so the parasites which can present recrudescence is Plasmodium falciparum and Plasmodium malariae. Okay, these are the ones which can present as recrudescence. So now we will move on to the pathogenesis of the parasite. So what is the most important feature about Plasmodium is that what happens is when the parasite moves in the endothelial cells, right, it gets sequestered in the blood vessels. So this is because of the presence of a factor that is called as Plasmodium falciparum endothelial membrane protein. Okay. Okay, so that is Plasmodium falciparum endothelial membrane protein, and also they will have a property of rosetting. Okay, they also have the property of rosetting, that is the infected RBCs will bind to the other RBCs, so that is called as the rosetting property. So this is the main pathogenetic feature of the malaria parasite. Then immunity. Immunity is some of the conditions which are inherently resistant to the malarial parasite. So what are they? So conditions like 1. Sickle cell anemia. Okay, like sickle cell anemia trait is resistant to it. Then fetal hemoglobin is resistant to the Plasmodium parasite infection. Then thalassemia trait is again immune. Okay, then people with G6PD deficiency is immune. Then Duffy coat antigen is also immune for Plasmodium vivax. So these are the different immune, the conditions which are inherently immune to the Plasmodium parasite. Okay, and some of the conditions like RBCs. Okay, if I talk about Plasmodium vivax, right, Plasmodium vivax will affect only the young RBCs. Okay, that is one of the reasons the Plasmodium vivax affected RBCs are bigger in size, because the younger RBCs are bigger. So if you know the developmental stage of RBCs, the reticulocytes are bigger compared to the mature RBCs. So that is why the vivax affected RBCs are bigger in size. Okay, this is also an important MCQ. The vivax affects the young RBCs. Okay, and now Plasmodium falciparum, they affect what? They affect all the stages of RBC. Again, this is the basis why falciparum is the most severe form, because that also affects the all the stages of RBC. That is why again it is the severe form. Okay. Now, so this is about the stages of RBCs that is infected by the parasite. Okay. So now we will go to the clinical features. How do the disease will present with? Okay, so if you talk about the clinical feature, the first and the foremost clinical feature any patient can present with is fever. And these patients can present with chills and rigors, followed by profuse sweating. It's very typical for malaria, right? So very typical for malaria is but at every cycle release of the merozoites into the circulation, they will start having fever, they will start having chills and rigors, followed by that they will have profuse sweating. This is a very common feature for malaria. Okay, fine. So what are the other features they can present with? They can also present with, in severe cases, they can present with anemia. Okay, and sometimes they can present with splenomegaly. Okay, so basically these are the common features. The other features are related to the complication of the disease. So complication if we take, so the complications are called as black water fever. When the malaria is very severe, it is more common with Plasmodium falciparum. Black water fever will be like there will be lot of hemolysis, and the patient will start, having the urine also as dark colored urine. So there will be a lot of hemolysis. That is the black water urine. Okay. And there is something called as an algid malaria. Okay. They can go for hepatosplenomegaly. They can go for hemolytic anemia. Okay. So these are the sometimes they can also go for, because of the severe condition, they can also go for shock. Okay. So that is one of the complications of the malaria that we can expect. So how do you now we go for the laboratory diagnosis of these patients? Yeah. And one more thing, I have forgotten to mention about the epidemiology. So in the epidemiology, if we talk about, India is one of the most endemic countries. And India, if we talk about which species is more common, in India, Plasmodium falciparum incidence is more common than Plasmodium vivax incidence. Okay, Plasmodium falciparum is more common than the Plasmodium vivax incidence. Right? Now we will go on to the laboratory diagnosis. In the laboratory diagnosis, we will take something called as a blood sample for malaria. It's very, very important. You will take a blood sample. From blood sample, we will proceed with something called as a smear. Okay. So peripheral smear could be thin smear or it can be thick smear. So what you do in a thin smear is you take one drop of blood, and then with the other slide, you have to just slide it over and to make something called as a tongue-shaped smear. So, okay, when you make the smear, the most important thing is it should have monolayer of cells to see the morphology clearly. Right. So, this is thin smear. Thick smear is you take 4 drops of blood and they are combined together like this. So, that is the thick smear. Okay. So, thick smear is basically used for the screening purpose. Just to know presence of the parasite. Okay. So, thick smear, we cannot do the species differentiation. Okay. So, thick smear, we can't do the species differentiation. Only with the thin smear, the species differentiation can be done. So, once a smear is made, the next step would be we will proceed to the Romanowski's stain. So, what will we do with the Romanowski stain? I have already told you Romanowski stain is a differential stain. Okay, the differential stain is something they will stain different structures in different color. Right? So, different structures in different color. So, now, I told you this. The RBC will be stained pink. Okay. So, the RBC will be stained pink. The cytoplasm will be stained blue and the nucleus will be stained red. So, it's very typical. Right? So, this is the Romanowski stain picture when you stain the parasite like this, right? So next here we will see that in a peripheral smear, what all could be the differences between the peripheral smear of vivax and falciparum. Okay, so the two most important species which is found in India is Plasmodium vivax and Plasmodium falciparum. So we will try to know the differences. Okay, so the first difference is, as we have already discussed, vivax is going to affect the young RBCs, whereas Plasmodium falciparum is going to affect all the stages of the RBCs. Okay, so if we talk about the young RBCs, it's only 2% in the circulation. right? It is only 2% in the circulation, whereas falciparum is going to affect all the stages of the RBC. That is why falciparum is very severe form. Okay, so that is why when we talk about falciparum, it is of the severe form. Okay, now the first thing is it is affects the young RBCs. So the second point is in the peripheral smear, when we talk about, we will find only the we will find all the stages in case of Vivax. That is the early ring stage, okay, late trophozoite stage, scion stage, and the gametocyte stage. Okay, whereas in the case of falciparum, we will find only the early trophozoite, that is the early trophozoite, that is the ring stage, and the gametocyte stage in the circulation. Okay, so it is very diagnostic. So when you find only the early ring stage and the gametocytic stage in the peripheral circulation, then it is Plasmodium falciparum. Now we will move on to the the discussion of what could be the differences otherwise in a peripheral smear. So I have told you, so this is Vivax and this is falciparum. So I have told you that, okay, so I have told you that Vivax, right, will have the early trophozoite to scion to gametocyte stage, everything present, whereas falciparum will only present with the early trophozoite, that's a ring stage, and the last stage, that is a gametocyte. So how do they differ? Suppose if I talk about the ring stage, what will be the difference? Number one, the infected RBC is bigger. Okay, the ring stage will occupy 1/3 the RBC, and then in the ring, the cytoplasm opposite to nuclei is thicker. I will show with pictures. Okay, will be thicker, and then the only one parasite will affect the RBC at one time. So whereas in falciparum, if I talk about the ring stage, one is the infected RBC can be same size as the other RBCs. Okay. Next, the cytoplasm opposite to the nuclei will be thin, and the nuclei can be divided, and same RBC can be infected by multiple parasites. Okay, and there can also be something called an aqual form in the falciparum. So what I'm talking about, let us draw and see it. So see that if this is the infected RBC of Plasmodium vivax. See that this is the infected RBC of, so I should go with the smaller size. So this is the infected RBC of the Plasmodium falciparum. I'm just drawing it because they will have very different forms. So when I'm talking about vivax, so I told you that the infected RBCs will be of bigger in size, and I told you that early ring stage there will be this ring, right? So I told you the ring cytoplasm, which is opposite to the nucleus, will be thicker. Yes, so This is the nucleus. So this is the common presentation. This is a typical presentation of Plasmodium vivax. So this is the RBC. This is the trophozoite stage of the parasite. So see, whatever you are seeing here, no, this whole thing is nothing but the parasite itself, which is dividing inside the RBC. Okay, so this is the, so what I'm talking about, so this particular thing, okay, the whole thing is the parasite itself. Okay, so this is the cytoplasm of the parasite, and this is the nucleus of the parasite, and this is called as an early trophozoite, or it is the ring stage of the ring stage. Right. Similarly, how this will present in the, and also you see proportionately the ring size compared to the infected RBC, it's almost 1/3 of the size. So when I'm going to talk about Plasmodium falciparum, you see the ring is like this. Okay. Like this. Like this. Okay, so this is the typical picture of the vivax. So what are all I am shown here to you? See, you can see one is the multiple infection, and here you can see the nucleus is divided. Okay, it is found in the different, so one nucleus here, one nucleus, so one nucleus itself is divided, you can see, and here also, like you can see, the same RBC is infected by two parasite, and this, the parasite cytoplasm is like merged with the cell membrane of RBC. This is called as an acole form. It's a very important MCQ. Acole forms are found in Plasmodium falciparum, and here again you can see multiple infection of the same RBC. Okay, so this is a very important question, very important MCQ. It is asked everywhere. You should know this. So now the next difference is, that the Plasmodium vivax can go into a late trophozoite stage, like it can go like this. So yeah, they can become like this, the late trophozoite stage, and then the cytoplasm can be distributed here and there, and then you might find the nuclei. Okay, so this is the late trophozoite stage. So further, this can go into something called as a scisont. So the scisont will be a proper thing inside the RBC. So actually you will not be able to distinguish the parasite and the the RBC membrane, because it has become bigger. And then you will find more of merozoites now inside the cell. Okay, so this is a scisont. You will have the merozoites inside the cell. So this is the scisont stage. So the last stage would be a gametocyte stage. So this is the gametocyte stage of Plasmodium vivax. So if you see, the gametocyte will be spherical. Okay, so this was the gametocyte stage. So in falciparum, the late trophozoite stage will not be present in the peripheral circulation. Okay, and the scisont will not be present in the circulation. Okay, and the gametocyte stage will be present, but the gametocyte of falciparum will be a banana shaped gametocyte. Okay, so they will be of something called as a Okay, so there will be a banana shaped or a sickle shaped gametocyte that will be present in the peripheral circulation. So these are the main differences which you will see in the peripheral smear. Okay, so now we'll see the pictures related to it. So now this is a peripheral smear which is showing the different stages of the parasite. So what is this? What is this stage? So this stage you can see that nucleus is there and the cytoplasm opposite to the nucleus is thickened. So this is the ring stage of Plasmodium vivax. Okay, and here this is the late trophozoite stage of P. vivax, right? And then the next thing here is you are seeing on the left side is you can see this one, no? This one here is the Acol form. So this one you can see a divided nucleus. This one you can see a small nucleus present. Okay, so this is the ring stage of Plasmodium falciparum. Okay, so now this is the scisont of Plasmodium vivax. And this is the gametocyte stage of Plasmodium vivax. You can see a round cell with condensed cytoplasm and nucleus. So that is the gametocyte stage. And this is a sickle shaped gametocyte is the gametocyte of Plasmodium falciparum. It is a very, very important MCQ. All your NEET questions and PGI questions and AIMS questions will be based on this peripheral smear picture. If you know this, then everything else is easy. So, you have done a peripheral smear from the blood. So, the next thing what you can do from the blood is you can do a quantitative Buffy coat, right? So, quantitative Buffy coat is you take the sample and then you put it in the quantitative Buffy coat and then see it under the fluorescence and then you will be able to pick up if there is any parasite. Okay, so for the quantitative Buffy coat to give the result, you will need around 3 to 4 parasite per microliter of blood. Okay, similarly, if I am talking about peripheral smear, okay, if I am talking about peripheral smear, the sensitivity of thick smear is around 5 parasite per microliter. Okay, if I am talking about thin smear, it is around 50 to 100 parasites per microliter I need for the smear to become positive. For QBC, I have told you already, it is 3 to 4 parasite per microliter of blood. If I am doing an immunochromatographic test, then I need minimum of 100 parasites per microliter of blood. So only if this much of parasite is present in that particular blood sample, then it will become positive. Okay, so for PCR, like less than 5 parasites per microliter, it will pick up the positivity. Right? So this is about the different sensitivity of different tests. So we are talking about the rapid antigen testing, that is the immunochromatographic test. Okay. So when I'm talking about the immunochromatographic test, here there are 2 types of antigen that are being detected. So if the first one is the immunochromatographic test which test LDH and aldolase. So this LDH will be common for all falciparum. If I have to do specifically for falciparum, then I have to do histidine-rich protein 2, that is for falciparum. Okay, this is a very important MCQ. Right. So now I can do the molecular methods also to detect the parasite from the samples. Okay, so now this is about the laboratory, and this sensitivity is also important. How much of parasite is required and how much of blood for the test to become positive is also an important question. So now we have diagnosed the patient is having malaria. So how do you going to treat them, right? So treatment of vivax malaria is very simple. Okay, so only problem with vivax malaria is it will present with relapse. So you have to keep it in mind when you treat the patient with vivax that relapse can occur. So we have drugs like, so some of them are sporocidal and some of them are gametocidal. So there is a drug which is called as primaquine, which is gametocidal. So primaquine has to be kept in mind whenever you treat the patients for malaria. Like suppose if it is Vivax, it is going to cause relapse. So you have to treat those patients with primaquine. So what is the treatment? You give chloroquine, okay, for 3 days along with primaquine for 14 days. Okay, so this is the treatment for Plasmodium vivax. So now we will go to Plasmodium falciparum. So when you treat Plasmodium falciparum, the most important thing which comes to your mind is drug resistance. So drug resistance is very common for falciparum malaria, and also the drug resistance is completely different when you compare the regions of India. So in the northeastern states, the resistance is a major problem. So the treatment of northeastern states is going to be different when compared to the other parts of the India. So when I talk about the other parts of the India, the treatment of malaria will be ACT combination. Okay, ACT combination. Okay, so what is this ACT combination? On day 1, you will give artesunate, okay, plus sulfadoxime plus pyrimethamine. Okay, so this is on the first day. On day 2, you will give artesunate plus primaquine. Okay, on day 3, it is only artesunate. Right? So if I'm talking about, so this is in the other parts of the world, other parts of the country for falciparum malaria. Now, when I'm talking about the northeastern states, it will be day 1. Okay, day 1, it will be artemether plus lumefantrine. On day 2, it will be artemether plus primaquine. And on day 3, it will be just artemether. Okay, so this will be the treatment regimen for falciparum malaria in India. So the other most important topic is vaccine for malaria. The licensed vaccine for malaria is RTS, okay, yes, AS01. Okay, the market name for this is Muscurix. So this is the thing which you have to remember about RTS AS01 is it is a genetically engineered vaccine. Okay, so it is a genetically engineered vaccine between the outer membrane protein of Plasmodium falciparum with the portion of hepatitis B virus. It's a very important question about the vaccine for malaria. The vaccine for malaria is RTS AS01. So this RTS, what is this AS01 is nothing but the adjuvant that is added to the vaccine. Okay, this AS01 is the adjuvant that is added to the vaccine is this. So that now we have completely discussed everything about malaria. We will go on to a next disease which is very similar to malaria, that is nothing but babesia. Okay, so babesiosis is a disease that is caused by Babesia microti. Okay. So when I'm talking about babesia, what is all is important? It is very similar to that of malaria. So the definitive host for babesia is tick, Isodex tick. Okay, and the intermediate host is obviously man, right? So the life cycle, if you see, so the tick bites man, right? And then it will inject the infective form into the man. That is the sporozoites, very similar. That will undergo the cycle in the RBCs. Then the merozoites are released. Okay, the merozoites are released. Then from the merozoites, it keep infecting the other RBCs and the cycle is repeated. Right, so this is about the life cycle of babesiosis. Right, so now here you have to remember, so clinical features is very similar. So they will present with fever, chills and rigors, and it can also lead to hemolytic anemia. Right, so now the diagnosis is peripheral smear, very similar to malaria. In the peripheral smear, a very typical feature is called as an Maltese cross. Very, 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 very important question. If you have a question which says that it is an intra-RBC pathogen and it shows a similar, very typical feature, that is Maltese cross, then the diagnosis is babesiosis. Right, so it is a similar life cycle as malaria. There is not much difference except for the typical finding of Maltese cross, and the vector is different, that is Isodex stick here, that's a definitive host. Okay, so how do you treat these patients? You give atoquinone plus azithromycin, or you can give clindamycin plus quinine. Is a treatment of choice for babesiosis. Okay, so now babesia is completed. We will move on to the next apicomplexa, that is Toxoplasma gondii. So Toxoplasma, where is it important? Toxoplasma, the definitive host is cat and the intermediate host is human. And please remember, the cat is the definitive host and the whole life cycle can be completed in the cat itself. It doesn't need human to finish the life cycle. Okay, this is human. So the morphological forms, if we talk about the morphological form, there are three forms which we should know. One is called as the sporulated. Okay, one is called as a sporulated oocyst. Another is tachyzoids. Okay, and the next is the tissue cyst, is also called as bradyzoids. Okay, it is also called as bradyzoids. Right, sporulated oocyst is the most common infective form. Okay, so this sporulated oocyst is the one which is excreted from the gut of the, the canines. So that when get ingested, then we will develop the disease. And the tachyzoids is mostly by means of blood transfusion or by vertical transfusion. If we have, then the trachyzoids are the infective form. So bradyzoids are the tissue cyst which will be like from undercooked food. If we have, then we can get. So all the three forms are infective forms. Okay, so one is the sporulated oocyst which is found in the soil, tachyzoids by blood transfusion or vertical transfusion, and tissue cyst is bradyzoids when you eat the undercooked meat. So all the three forms are infective. Right, so what is the next thing? The mode of transmission. So mode of transmission also I have already discussed. So it can be fecal-oral, oral by ingestion of the sporulated oocyst, or it can be vertical, right, or it can be by the consuming undercooked meat. Right, fine. So when we talk about the life cycle, so it is very clear here. So this is the fecal oocyst that is being fecal-orally taken by man. That is one of the ways. And then when this cat lays this oocyst in the environment, so some of the animals like pig and cattle can eat this, and then the larval forms, that is the bradyzoids, can get deposited in them. And when man eats the undercooked meat of them, then it can get transmitted, or from one person to another when there is a blood transfusion, again the blood form, that is the tachyzoids, can get transmit. So and this particular cycle will keep continuing in the environment. Okay, so this is the life cycle of the Toxoplasma. The very important point is the most common transmission is sporulated oocyst. So sporulated oocytes gets inside the man, it goes as tachyzoids. Okay, so and then in the blood, it goes to the tissue and gets deposited as tissue bradyzoids. Okay, so and then it gets deposited in the various tissue, and that is what is causing the symptoms. Okay, fine. Okay, we'll go to the next point of discussion. So the clinical features, right? So if we talk about the clinical features, mostly in the adult, they are mostly asymptomatic until the immune system functions properly. Okay. Next is the patients can present with one common symptom, that is the lymph node enlargement. Right? In congenital toxoplasmosis, so the more severe form, suppose if the transmission occurs in the first trimester, will be the more severe form, right? Or when in the third trimester, if the transmission occurs, the severity will be less, but third trimester have more chances of spreading. Is in the third trimester, but the severity would be less. So what to remember in this? There is a classical triad of congenital toxoplasmosis is chorioretinitis, okay, then hydrocephalus, okay, and the cerebral calcification. Okay, so this is the classical triad: chorioretinitis, cerebral calcification, hydrocephalus, and they can also present with mental retardation and microcephaly. Right? So these are the congenital toxoplasmosis features. And in HIV individuals, what happens is it will be disseminating. Okay, in HIV individuals, the most common would be CNS. It will cause encephalitis. Okay, and the most common site would be brain. Right? So now we will move on to the laboratory diagnosis. So in laboratory diagnosis, you can take a blood sample to look for the transient presence of tachyzoids. Okay, and also you can take the tissue biopsy to look for the bradyzoids. Okay, fine. And then in the blood, you can look for the presence of tachyzoids, and the organs you can look for the bradyzoids, right? So now in the blood, you can do an other test. Okay, so in the blood you can also look for the serology, just to know, because toxoplasmosis is a chronic condition and patient will suddenly start presenting the symptoms when the immune system goes down, and this scenario is very, very important when the person is pregnant. Okay, when the person is pregnant, this becomes of importance, just to know whether it's a current infection or a past infection, to prevent the transmissibility. So in that case, so you look for the IgG in the serum. So if IgG is there, then it is a chronic infection. If IgM is there, it is a acute infection. And you look for the avidity of the IgG binding. If the avidity is less, that means it is a recent infection. So because it had a recent infection, the binding capacity has become less. So IgG avidity test, if it is lesser, that means it is a recent infection. Okay, what is the Sabin-Feldman dye test? It is a complement fixation test. Here what you do, you take the patient blood looking for antibodies, then you take the complement, then you put the live tachyzoids, and then you put a dye, and then later you see if the patient's serum is positive for antibody, the tachyzoids will be distorted. If the tracheozoids are distorted, that means the patient sample is positive for antibody, right? So then you can go for the detection of specific antigen in the patient, right? And then you can go for molecular detection of specific targets, right? And you can also go for animal inoculation, that is the mice. So in the mice, you can do the animal inoculation. Okay, and congenital toxoplasmosis, again, you have to look for an IgM or an IgM or an IgA antibody. If it is present, that means it is the infection of the infection in the baby. If IgG is present, it is not conclusive because IgG antibodies will be transferred from mother. Okay, so to say congenital toxoplasmosis, you should be able to see IgG and IgA and IgM in the baby, then it is congenital toxoplasmosis. So how do you treat congenital toxoplasmosis? Yes, pyrimethamine plus sulfadiazine with folinic acid for 1 year is the drug of choice, right? And for pregnancy, the drug of choice is spiramycin. For pregnancy, the drug of choice is spiramycin. For HIV, cotrimoxazole is the primary drug of choice. Okay, so next we will move on to the next topic, that is the other apicomplex of importance is Cryptosporidium, Cyclospora and Cystisospora. Okay, so now if I'm going to talk about it, okay, so now the infective form for all the three is sporulated oocyst. Okay, so the infective form would be sporulated oocyst. Okay, so this will be sporulated oocyst for Cryptosporidium, unsporulated oocyst for this, and for Isospora again. Okay, so the infective form would be the sporulated oocyst. Sporulated oocyst. Okay, so the diagnostic form is again for Cryptosporidium, it will be sporulated oocyst. It will be unsporulated oocyst for Cyclospora, again unsporulated oocyst for Isospora. Okay, that is the diagnostic form. The oocyst size is of very important because it is from here. If you see, Cryptosporidium will have a size of around 2 to 5 micrometer in size, whereas Cyclospora is just double the size. It will be around 4 to 8 micrometer in size. Isospora will be bigger. It will be around 15 to 20 micron in size. Okay, so these are the different sizes of the oocyst. Right. Next, so the oocyst shape. If you talk about the oocyst shape, so this will also be spherical. This will also be spherical, and Isospora will be little of oval shaped. Okay, so this will be the shape. So sporulated oocyst, if you see, it will be of, I have already mentioned you regarding the size. So it will have 4 sporozoids inside for Cryptosporidium. For Cyclospora, I'll put a bigger size. For Cyclospora, okay, it will have 2 sporocysts inside and with 2 sporozoids. Okay, and for Isospora, okay, Isospora, again it will have 2 sporocysts and it will have sporozoids each. Okay, so this is very typical. So it will have 4 sporozoids. It will have 2 sporocysts with 2 each sporozoids. It will have 4 sporocysts with 4 sporozoids each. Very right. The next difference would be the acid fastness. So if you talk about acid fastness, then it is this cryptosporidium will be uniformly acid fast. Cyclospora is variable acid fast, and cystisospora also will be uniformly acid fast. Okay, all the three are acid fast, but cryptosporidium and isospora will have uniform acid fastivity. Cyclospora will have a variable acid fastness. Okay, autofluorescence is a very, very important property. Like, okay, if I talk about autofluorescence, we talk about the autofluorescence. Okay, if I talk about autofluorescence, autofluorescence is positive for cyclospora, and cryptosporidium will have no autofluorescence. Then it will be autofluorescence present for cyclospora, and for isospora it is plus minus. It might be there, it may not be there. Remember, in the cycle you will have light, so it fluoresces. Remember like that. Okay, so cyclospora has autofluorescence property, right? Okay, so now if I talk about the treatment option, okay, the treatment for cryptosporidium is nitazoxanide. For cyclospora it is cotrimazole, and for cystisospora also it is cotrimoxazole. The very important point about cryptosporidium, cyclospora and isospora are they causes persistent diarrhea in HIV patients, and if you do the acid fast staining with 0.5 to 1% sulfuric acid in the stool sample, they will show acid fast oocyst. Okay, so this is about the apicomplexa. So this is about apicomplexa. Thank you.